That'll do it. Sorry about that to everyone on Zoom. All good now, I'm assuming? Yeah, we'll move on anyways. Someone else didn't read. Anyways, okay, I'll move on. Hopefully people can hear me.
Yes, it's fine now.
It's all good. It always happens on Zoom, doesn't it? All right, so yeah. So for the people that weren't listening, I was just going over some of my honors research, which was in the Strzelecki Desert. So yeah, what I was doing, I was part of a long-term monitoring study looking at how the dingo can affect all sorts of animals and plants and ecosystem functions. So what we would do is we would capture like a whole range of animals. So things like these goannas and some snakes would accidentally fall in a pit trap too, but also a lot of rare mammals that you don't usually see. So these ones down here are dusky hopping mice, which is listed as a vulnerable species. And then this thing up here being released is called a mulgara, which is like a small rodent-sized marsupial predator, which is quite hard to find. But yeah, all of these animals are obviously really cool, quite photogenic and charismatic. But the thing that I was most interested in were these really large, scary, freaky macroinvertebrates that most people sort of hate, but I don't know, I find them quite interesting. So yeah, in the desert, there's, for whatever reason, these really, really large species of invertebrates. These big whistling spiders, it's sort of like a tarantula sort of thing. Big scorpions, very large centipedes, also really large beetles too. So I was looking at how the dingo can possibly influence these insect communities. So if you didn't know already, the dingo is a really important sort of keystone predator in Australia's ecosystems. So what they do is they exert sort of top-down effects on things. Like if you think about what a dingo does, it eats kangaroos. Kangaroos eat grass. So if you get rid of dingoes, there'll be a proliferation of kangaroos and probably a lot less grass. So we're looking at sort of these keystone interactions. As an ecologist, I think they're pretty cool and important, but not everyone sees it that way, which has sort of manifested in the construction of the Dingo Fence, which is this 2-meter tall fence. It's one of the longest man-made structures on Earth. And it essentially runs, well, it is, yeah, it's literally one of the longest man-made structures on Earth. And it runs pretty much from the coast of Queensland, intersects the entirety of the continent, all the way down to South Australia and then a bit out into the ocean too. And what it does, it excludes dingo from grazing land inside the fence in New South Wales, but then outside the fence, dingoes are really common. So what this does, I mean, it wasn't the purpose of constructing this fence, but what it does for an ecologist is it sort of sets up a natural experiment. So what we can do is we can hop on either side of the fence. One side doesn't have a dingo, one side does have the dingo, and then we can just like compare the communities and just see, you know, what animals are present when you have the dingo, and then what animals sort of go extinct when you get rid of the dingo. So yeah, well, we were mostly looking at these things called trophic cascades, which is sort of what I mentioned a little bit before. It's where like an apex predator has like flow-on effects to things lower down in the food chain. So if you think about the dingo, what it does is it both eats and scares away a lot of invasive cats and foxes. So what happens when you have big populations of the dingo is you won't have many cats and foxes. And cats and foxes then eat smaller things like native insectivores. So species that eat insects, like goannas, bilbies, some of those things that I showed before, like mulgaras will eat a lot of insects as well. And what this does is indirectly, it means that when you have lots of dingoes, you have a lot of insectivores because you've gotten rid of all the cats and foxes that are eating the insectivores. So what my research was for honours was just to see if this has like a flow-on effect to the insect community. So yeah, when you throw up this big dingo fence and you get rid of the dingo, you have a lot of cats and foxes and you have virtually no insectivores. And this is exactly what we were finding in our study system. So if you go on one side of the fence where the dingo is absent, it's really hard to find goannas. They're still there, but they're in really, really low numbers. Mulgaras are pretty much absent. Native mice are also quite absent too. But when you hop on the other side, they're really abundant. And this is just like within 10 kilometers of each other. The only difference is that there's a fence running down the middle of this area. It's a really homogeneous area, so there's no differences in habitat. It's just rolling dunes for like tens and tens of kilometers. The only thing that's different is just one side has the dingo and one side does not have the dingo. So yeah, looking at how it affected insects, outside of the fence, I was finding a really big abundance of these things called pie dish beetles, and also native cockroaches as well. And I think the reasoning behind this was that, as I mentioned before, when you have big populations of the dingo, you have a buildup of vegetation, and these two groups like to consume decaying organic matter. So outside of the fence, there were a lot of these two groups. And then inside of the fence, there was an entirely different insect community. And I was finding these really big predatory centipedes and grasshoppers, which are really tasty for a lot of insectivore species. So I think the fact that there were no insectivores on the inside of the fence meant that these two species were sort of released from predation, and they became a lot more abundant. So yeah, what I liked about all this research was obviously the field work, but on top of that, it sort of opened my eyes a bit as to how you can study insects. So for instance, this project wasn't set up to actually study insects. It was mostly focused on the dingo, but we were able to weave in this really interesting story about how, this big species that doesn't really interact with insects can actually sort of change the communities indirectly. But yeah, I'll move on now to what I'm currently studying and also what your society is partly funding. So the focus of my PhD is on invertebrates and microbes that live in the leaf litter. So a really, really overlooked and undervalued group. So this is where a majority of terrestrial diversity really sits. So in this leaf litter layer, but we just like step on it and don't really dig around in it to actually see what insect species are there. So they don't get a lot of attention in comparison to like pollinators and things that are quite easy and sort of vibrant with color. But yeah, what I was trying to do was I was trying to rewild invertebrate and microbe communities to both improve diversity and also ecosystem function. And for anyone that's not familiar with the term rewilding, it's just a fancy word for reintroductions. So what we were trying to do was to reintroduce invertebrates and microbes into areas where they have become absent, in hopes of improving their diversity and then also improving ecosystem functions as well. And we're doing that on farms, so privately managed farmland. This is an example of one of my farms, a lot of invasive grass, like pasture species like that, and just like an abundance of sheep. So this is like high density livestock farming. So yeah, as everyone probably knows already, a major cause of species decline is land clearing for agriculture. But a lot of farmers are quite proactive in trying to reverse this. So what they will do is they'll like put a side of the farm, like an extra pasture, and then plant a whole bunch of tree species in it, in the hopes of growing back a native forest in an area where there wasn't a native forest, or it hasn't been a native forest for a very long time. So what they'll do is they'll go out and they'll plant a whole bunch of tree species, grow a really big, beautiful, mature forest like that. And the hope is that all these animals from these surrounding remnant areas can then recolonize this new habitat, such that the remnant species can also be found on the farm. So it's trying to get the same sort of communities between these two areas, and just sort of increase the habitat range for a whole bunch of remnant species. But yeah, it's not always straightforward, unfortunately, so it's not as simple as that. One of the reasons behind that is a lot of these farms are revegetated in small sections, so clumps of restoration, surrounded by these big pastures, which are quite hard for animals to move through. So they sort of exist as like an isolated habitat island, is the way that I like to think about it. And if you compare it to like an island in the sea, so if you have an island far off in the ocean, it's often quite hard for animals on the mainland to actually disperse to that island and recolonize it. Only things that are really good at moving around can actually reach these little habitat islands. And again, if we visualize it like this, so we have our remnant sites like that, really big swath of pasture that's hard for things to move through, and then an isolated pocket of revegetation like that. Things like birds, which are really good flyers, they can easily move from site to site, don't find too much of an issue with this. So you'll find that birds from these remnant sites can easily fly into these revegetation sites, such that they have the same sort of community and the same levels of diversity. But when we start to look at invertebrates, often quite small, many don't have wings, really hard for them to move from space to space. And what this results in is less species recolonizing these revegetation areas. And because these species are so important for ecosystem function, you can get less efficient ecosystem functions and health as well. And this pattern sort of holds for microbes too. Like, not a lot of people think about this, but microbes also differ in their ability to move from space to space via spores. So, you know, the further the distances, the harder it is for a lot of microbe species to actually recolonize these patches of habitat. And yeah, this is exactly what I find in my study system. So when I sample on revegetation sites, they often have a lot lower levels of diversity as compared to some sort of nearby remnant site that's quite close. So this is where rewilding comes in. This is how we're going to fix it. So what we do is we essentially go into these biologically rich remnant sites with all these, you know, diverse species communities. We scrape off a bunch of leaf litter, and within that leaf litter is the whole community. So there's like hundreds of species of invertebrates and thousands of species of bacteria and fungi. We literally just pack it into a truck and drive it onto the farm and then just dump it into the revegetation site. It's not very technical or difficult. You know, these things are quite small, very easy to move around. It's not a dramatic like rhino release or anything like that. It's just dumping leaf litter. So yeah, that's what we do. That's our actual rewilding sort of event. And we're hoping that we're going to reintroduce some of these species that are struggling to get to this area, thereby increasing diversity and ecosystem function as well. I hope people can see that. It's a bit small with the writing, but these are my field sites. So I'm working in the Strathbogie Ranges, which is about 2 hours north of Melbourne, in a really heavy agricultural area. So it's been extensively cleared for hundreds of years, but it still has these patches of remnant sites down here. And within my field sites, I have 3 treatments. I have a remnant site, which is the source of my leaf litter and also contains my like ideal communities. So what I want my invertebrate and microbe communities to look like. I have a rewilding transplant site, which is where we actually do the leaf litter transplants. So that's one of our treatments. And then I also have a control revegetation site, which has had nothing done to it essentially. So no transplant at all. And obviously we just include that control just to make a comparison to our transplant sites, just to make sure that there's actually been a change in the species diversity. And yeah, that's how some of my sites look. So again, you know, a bit of revegetation there, and these big sort of swaths of invasive pasture, which is quite hard for things to move through. A remnant site up here, so like a conservation park, I think that one is. And then all these big sort of expansive pastures, and then a transplant site down there. So you can see how for these small things, it's quite hard for them to move around this really highly fragmented area, which is why we're doing this whole leaf litter thing. So yeah, because we focused on the entire community, there's like heaps of things that we can look at. Obviously, we can look at the microbes, which are the bacteria and the fungi. We can also look at the macrofauna, which are the bigger things like the cockroaches and the millipedes, things that are quite easy to see with the naked eye. But another group that people like almost never think about or sort of know about is this group called the Meso Fauna, which are things that are usually 2 millimeters in size. A lot of things like mites and springtails. They're like the cornerstone of many ecosystems in terms of diversity. So this is where the bulk of diversity is in many terrestrial systems, these really diverse communities of mites and springtails, and they're really important for actually breaking down leaf litter and cycling it back into the soil. So they're really important for soil health. But if you thought these were overlooked, these are very overlooked in terms of what people generally care about. So I was with a springtail taxonomist, and she was saying that almost half of the species that I presented to her were undescribed to science, and she's been doing it for like 70 years or something like that. So it's a really understudied group and really overlooked. But yeah, within the mesofauna, there's also a lot of insect species too, like a lot of beetles and wasp species can be less than 2 millimeters in size. But yeah, I won't go into results for all of these groups. I'll try to focus on the winners because not a lot of things actually survive the transplant, unfortunately, likely due to the fact that remnant sites and revegetation sites are just inherently different. So remnant sites are quite moist, whereas remnant sites aren't as moist. And a lot of these animals rely on moist leaf litter to actually survive. But there's been some really clear winners, and it's mostly restricted to these really tiny little beetle species. So looking at species richness, which is just a count of how many species you can find in each treatment, just going up this way. I did a baseline measurement, so that's before rewilding. And you can see in terms of beetles species diversity, the control and the rewilding transplant site had really similar levels. But then there's this really sort of dramatic increase in the number of beetle species that we could find in our rewilding transplant site after rewilding. So we have more species than the control, which is good. But we also have the same number of species in the remnant, which means it's really likely that we've introduced a whole bunch of remnant species into this area that wouldn't have got there otherwise. And then when we drill down into what species were driving that change, I was finding that these little things called rove beetles were a lot more prevalent, which are, I mean, some of them are quite big, but the ones I was looking at were about 2 millimeters in size. And they're really important predators in this ecosystem as well. And for whatever reason, they were just really responding to our transplant treatment. So yeah, what else can we look at with this stuff? Well, because invertebrates cover spectrum feeding groups, so they're really important for every ecosystem. They can obviously be pollinators, herbivores, carnivores too. What we were interested in was, have any of these trophic groups responded from rewilding? Have they increased in abundances or diversity? And amazingly, we found that the number of predatory species increased. So again, this is just a count of the number of predator species you can find in each of these treatments. And moving from before to after, you can see that same pattern of the rewilding transplant site having many more species in comparison to the control. And a lot of this is probably due to those rove beetles that I mentioned previously, which are predators. But I also found that a lot of predatory mite species increase in abundance too. So these are things like red velvet mites, which people might know about. And then these other things called snout mites, which people probably don't know about. They're really important predators. So you can see one is eating a springtail right there. So yeah, these are really, really fundamental components of the ecosystem, which actually keep these really hyper-abundant herbivorous invertebrates in check. So it's good that we're seeing an increase in the number of predators. So yeah, how does this all link back to ecosystem health and function? It's a bit of a gross picture. But what I was focusing on was the microbes and invertebrates in the leaf litter. And these groups are really, really important for the actual process of leaf litter breakdown. So they'll actually consume and then recycle the leaf litter back into the soil as nutrients. So it's this really important but often overlooked cycle of nutrients from tree to leaf litter, and then broken down back into soil as nutrients, and then absorbed back up by a tree to use for growth as well. And this is obviously especially important on farms because a lot of farmers are interested in how healthy their soils are. So they want to have really nutrient-rich soils. So having really efficient levels of decomposition is quite important for a lot of these farmers. So yeah, our question was, you know, if we've seen these changes in species diversity, have we also seen changes in ecosystem function? So have we increased the speed of leaf litter breakdown? So leaf litter breakdown is a really, really complex process, as I'm finding out. It's both driven by invertebrates and microbes in tandem, so they can work together to break down leaf litter, and they can also compete with one another too. And then on top of that too, there's all these abiotic variables that can also influence litter breakdown. So I pretty much looked at all of these just to see what was actually driving leaf litter decomposition in my study system. And we do that via these things called leaf litter bags, which are just essentially bags stuffed with leaf litter, as the name sort of suggests. And what we do is we weigh them, and then we leave them in the field for 3 months, and then we bring them back, and then we weigh them again. And the mass loss from that leaf litter bag indicates how much of it was actually consumed in the field by invertebrates and microbes. So the higher the mass loss, the more efficient the speed of breakdown is. And amazingly, we found that rewilding increased decomposition rates. So this is just mass loss here. The higher the level, the more mass loss. And as you can see, leaf litter was breaking down faster in the rewilding transplant as compared to the control when we measured it 3 months after the first rewilding event, which occurred in November 2018, I believe. And I did this sort of fancy analysis and found out that this was mostly driven by the abundance of arthropod detritivores, so things that actually consume the leaf litter, like millipedes, isopods, cockroaches, that sort of thing. And then I measured it again 18 months after rewilding and found that there were no differences, which was probably due to really moist conditions. This is during, I think, a winter sampling session. So really moist conditions across the board, but where sites had higher temperatures, that was driving a lot of the leaf litter breakdown. So decomposition wasn't actually being driven by invertebrates, but more by the environmental conditions. And then I measured it one more time, 36 months after rewilding, and found like a really dramatic increase in decomposition in the rewilding transplant sites. It even supersedes the remnant too. And again, when I did this fancy sort of analysis, I found that this was driven by the abundance of arthropod detritivores. So if you see, this is the leaf litter as it looks like when I leave it out in the field. So we tend to go for leaves which aren't damaged at all. And then after 3 months, when I brought them back, they were essentially torn to shreds by something quite large, which indicates that something like a beetle or a cockroach or a millipede has come through and just absolutely smashed the leaf litter. Excuse me. But yeah, a really cool pattern, I'd like to think. So yeah, just to summarize my PhD so far, I'm still in the process of writing it up, but I think that's quite a few results. Leaf litter transplants have increased on-farm diversity of beetles. That's pretty cool. It's increased the speed of decomposition, which was a really amazing thing to find as well. And decomposition is largely driven by the abundance of invertebrates, which doesn't always happen in ecosystems. So that's also quite cool to see. But there's a lot more things that we can look at. So as I mentioned, we've transplanted the whole community. So we've transplanted all the microbes, all the invertebrates. There's a lot of different studies that we can do with this. So I'll briefly chat about some preliminary results because I think they're kind of cool and I kind of want to share them. So what I'm working on now is this thing called a microbiome study. So just to set the scene, every complex organism like an invertebrate, and everyone in this room has a really diverse assemblage of bacteria in their gut and on their skin, which is called their microbiome. And these microbes are really important for the host of the health. So they'll provide some sort of benefit to their host, and in return, they get a safe place to live. So for instance, when you think about like a termite, they consume wood pretty much entirely, but they wouldn't be able to consume wood if it weren't for this really diverse community of microbes in their hindgut. So these species are entirely dependent on these microbes. So yeah, what does this have to do with rewilding? Well, if you think about what we did, we transplanted all the microbe community. So it's likely that a lot of these different species of bacteria and fungi established in the new transplant sites, which presents a new community for invertebrates to then interact with and potentially incorporate into their own personal microbiome. So what we can do is we can look at whether or not leaf litter transplants have changed the microbiome of invertebrates. Is their microbiome more diverse? And importantly, has this new microbiome potentially provided an advantage for the invertebrates? Like, is it enabled them to maybe consume a different type of food or something like that? So yeah, I'm looking at 2 species of interest. I'm doing like a native versus an invasive sort of comparison. My native species is the honey brown beetle, which is mostly present in Southern Australia. And then my invasive species is the Portuguese millipede, which is really prevalent in Southern Australia, but I don't think it has reached Sydney yet. And what they do is they form these really big sort of masses of individuals after rain. So they don't actually eat crops or anything, but they're technically listed as an economic pest just because they form these really big sort of swarms. And there's like a story going around that they caused a train crash in WA like 10 years ago because they massed on the tracks, and when a train tried to stop, it squished them all and there was no friction and caused a train crash. But other than that, they don't really do anything. They're just sort of annoying. Like they'll mass in your house or something like that, but they don't actually eat crops or anything. So that's quite good. But yeah, I'm doing like a comparison between these 2 species, mostly because they're really abundant in my study system, but also they're detritivores, so they actually consume the leaf litter. So the microbes that they have in their microbiome is really important for that process as well. And to do these microbiome studies, what you do is you essentially like take an invertebrate, you mush it up into like a slurry, and then you extract a bit of the bacterial DNA from that and then sequence that bacterial DNA. So I ended up mashing up about like 100 individuals from each of these species. Yeah, I haven't had a chance to do a deep dive into this data. This is sort of the first time I've really shared it with anyone. But from what I can see, it looks really promising and interesting. So this is just a count of bacterial species. So the number of bacterial species in the microbiome of each of the invertebrates. And when you look at the invasive millipede, you can see that there hasn't been much change at all across any of the treatments. But then when we look at the microbiome of the native beetle, you can see that for whatever reason in the rewilding transplant site, it's just like gone gangbusters with the amount of diversity of bacterial species it has within its microbiome. And I'm not entirely sure why this would be the case, but that's sort of what I'm going to go and explore now, just figure out what bacterial species are driving this change and what sort of effect it's had on the species as well. But yeah, it's a really strange pattern, and I can't really explain why that is happening, but I'm not a microbiologist by trade, so I'm going to do a lot of background knowledge on theory and stuff to figure out what's going on there. But yeah, just on like a final sort of sobering note, insects are declining globally, still at a really alarming rate. Not entirely sure why, in a lot of cases, why they are declining. They're even declining in national parks and stuff too. So it's sort of like an across the board sort of issue, which is why projects like mine are really important, because I've hopefully shown tonight that it's not that hard to actually increase diversity on all of these managed farms. And then even though that like a lot of the things that I'm working with aren't necessarily cute or cuddly or particularly photogenic to a lot of people, they're really, really important for both diversity and function, both on farms and in wild areas. So it's really important that we keep sort of learning about them and, you know, investing our time and money into figuring out how we can sort of stop these declines. But yeah, thank you so much for that. And thank you again for coming out and providing me with a bit of funding for some of this research. Cheers.
Do you want to pick the lights on, please? Wonderful.
Oh,
extremely interesting.
Oh, cool. Awesome.
Yeah, I'm sure there's a lot of questions going forward.
Yeah, I hope it makes sense.
Yeah,
sort of stuff
I'll just move closer to the figure. Are there any questions coming in on the chat line, Graham? We'll start. Deb, you're always first to lead the charge.
Sounds very good.
Yeah. Come in and speak into the microphone if you don't mind.
Okay, thanks. Really interesting talk. You talked about the leaf litter and the moisture being an important function in influencing your invertebrates. Does leaf litter depth make a difference to your invertebrates, and does that increase with age of your remnant? Is there a relationship there between that?
Yeah, that's a really good question.
A second question, if I could slip it in, is when you're looking at the DNA of your microbes, how do you get just the DNA of microbes and not the rest of the organism and things?
Oh, it's really complex.
I'll go there.
I'm not a geneticist, but I'll try and explain that one. The first question is pretty straightforward. It's about the depth of the leaf litter. Yeah, so this stuff is considered food to these invertebrates, but it's also considered habitat. So they're living in their own habitat, which they're consuming. So the more leaf litter, the more invertebrates and microbes in terms of diversity. So depth is really important. So I actually go out and I measure depth and just make sure that depth is the same across sites, just to make sure that that hasn't influenced any of the results. But I often find that depth is higher in the revegetation sites for whatever reason, potentially because it's not breaking down as fast, or just the species that they planted just might shed more than in the remnant sites because it's a really mature forest. But yeah, depth is very important when you're talking about litter, invertebrates and microbes. Second question about the bacterial and how you ignore everything else. So we use specific primers that latch onto just bacterial DNA. And then on top of that, we do these sort of like cleaning and filtering steps, which get rid of non-target DNA. And then on top of that, we do like bioinformatics and definitely filter out everything that isn't a bacterial species. So we're pretty sure that what we're looking at is bacteria and not something else. But yeah, a geneticist would be able to explain that, I think, a lot better. But yeah, it works essentially. Yeah.
Yeah. Sorry. Tony, I meant. I must have been looking the other way.
Hi, that was really interesting. So it was really, really interesting. Just one thing that occurred to me, how do you cope for the fact that the composition of the remnant forest might have different trees, different bushes to the rewilding area and to your control area?
Yeah.
So do you do that or not?
Yeah. Yeah. So the question was, how do we factor in the fact that the habitats are just inherently different? So we're sort of restrained by what species they planted 30 years ago, because all these revegetation sites are about 30 years old. But one thing that we do is we just use replicates. So we just use like 6 different sites just to sort of even out any of that difference. And fortunately, in my area, the overstory species are really dominant. So it's only like 1 or 2 species of eucalypt that actually sort of dominate these remnant sites. Yeah, I've factored into the analysis that it's not the most important thing. The most important thing is like the leaf litter depth and the moisture. But yeah, it is something to think about for sure. Yeah.
Anything coming in? Anybody out there in Zoom land would like to ask a question? You could unmute yourself and ask a question. No? Graham?
I was wondering, you were saying how important the leaf litter is to invertebrates. What happens when a fire goes through? When it takes out all the litter?
Yeah.
How does the bacteria recolonise or re-establish itself?
Very, very, very pertinent question. Very pertinent for obvious reasons with the megafires. But I'm also part of a lab that is specifically looking at the effect that the megafires have had on leaf litter dwelling invertebrates, not so much the microbes. And we've had a lot of PhD students look at that too. And if I'm just like recounting what he found, it was that it depends on the species. So some species of invertebrate, when a big megafire comes through, they'll just dig down as deep as they can and just try and wait out the fire. And like amazingly, a lot of them actually survive like a mega fire coming through. Or they might like try to bury under a log, because logs in their core, it doesn't exactly get affected by the fire. And then some species will actually just be able to disperse back in. So they'll just perish, and then patches hopefully around the area, they'll fly from there. So it just depends on the species, but they do have a massive effect, especially for things that can't fly very well and can't disperse very well. Which leads me into the other project that my lab is looking at, is we're doing these litter transplants into these megafire spots. So we've actually taken leaf litter from some of the fires, some of the areas where the fire hasn't affected. And we're doing this in like around Malakuta and the sort of southern area of Australia. And then we're transplanting it into these really severely burned sites. And we're just seeing if that has sort of assisted the recolonization, essentially. So yeah, it's a good question, but yeah. Some of them die and some of them don't, essentially. Yeah.
Right.
Very interesting.
So my question is, how fussy are the little critters that are eating all this stuff? Will they eat non-native leaves and non-native leaf litter, let's say, as well as the native stuff? And within the native stuff, how fussy are there? Some that like Banksias or eucalypts or whatever.
Yeah. It's a good question. How fussy are the invertebrates? In terms of breakdown, I think acacia breaks down pretty well in comparison to like eucalypts. So for the eucalypts, they don't break down as well, and they have, I think, some more volatile compounds which are harder for things to consume. But in terms of fussiness, the short answer would be we don't know, because no one really knows much about these species anyways. Like, they're not really described as science, so we're not entirely sure what they're eating in all of the instances. But for the most part, I like to think that these things aren't particularly fussy, and they'll just eat whatever's been given to them. A lot of the things that we look at eat the microbes too. So they'll actually graze on the microbial species. And then in this area that I'm in, it's an agricultural area. So we have like a lot of invasive invertebrates too, which I think are particularly not very fussy at all. And they'll just eat whatever they can get a hold of. But yeah, short answer is I'm not entirely sure, but yeah.
Yes.
Peter, I'm wondering if you have had any experience on these, this is farmland that you're working on, the use of agricultural chemicals either through stock dips and drenches or spraying. Have you been able to exclude that from the sites that you're working on, or do you know what effect that might have?
Well, fortunately, the farmers that we're working with are quite environmentally switched on. I mean, they've taken the effort to actually put apart part of their farm to revegetate. So they're quite interested in getting diversity back. So I'm not entirely sure that any of them would do any sort of particularly bad spraying. A lot of them are really interested in the project. So they work with Landcare, these farmers. And whenever they see me, they try to like pull me over and say, you know, how are the invertebrates going now? Are you finding results and that sort of thing? But there are like canola farms bordering some of my sites, which could potentially, I don't know if canola gets sprayed particularly heavily or not, but that could potentially be a factor, but it's something that I have not incorporated in, but it is, yeah, an interesting point to think about. Yeah.
I'm just thinking about if you wanted to, if you wanted to rewild a whole site, how much leaf litter would you have to bring in? Like you put a spot there, how much travel is there of the microbes and the organisms from one bit to another, you know, to go through the whole, the whole site?
That's a good question. So I didn't want to get into the nitty-gritty methodology because it's quite boring, but what we do is we have this 10 by 10 meter sort of transplant grid. So that's where we actually do the transplants, and we take like 80 centimeter by 80 centimeter samples of leaf litter, and we take 10 of those, and then we dump 10 of those into the 10 by 10 meter grid. And then we sample 2 meters outside of the grid for our invertebrate communities. So we can definitely know that they've at least traveled 2 meters. But the idea is that they should proliferate through the entire revegetation site if they can make that 10 meters. And we've also sampled 10 meters from this grid, but it's been too difficult to sort of process all of those samples. But we at least know that they've traveled 2 meters, which is good. I can assume that they've gone further than that, but yeah, once they're there, they should be able to spread out as much as possible. Yeah.
Nothing coming in on the chat line. Back to Deb. Come up.
Peter, do you have any? Yes. Do you have any in fauna in your remnants that are involved with turning the soil, like lyrebirds or bandicoots or potoroos, bush rats and things? And if you do you think that's do you need to think that they need to be transplanted as well?
I think everything should be transplanted. If it's not there, you should put it back. I mean, if it used to be there and humans got rid of it, it should be put back to where it was. I have a colleague who works exclusively on lyrebirds, and they're obviously really important ecosystem engineers, so they turn over a lot of soil. And they're not actually on this side of the Strathbogie ranges. So for whatever reason, he thinks that they were absent there from about 25 years ago, but they're sort of around the area. But just from looking in my remnant sites, I don't see much evidence of scratching and turning over. There's a lot of feral deer. I mean, they're probably doing some bad stuff and trampling, but in terms of small ecosystem engineers actually moving the soil, there'd be not potoroos. Maybe some bandicoots, I'm assuming, but there's not a lot of evidence of a lot of digging. It's a really fragmented sort of area. So my remnant sites are quite isolated and there's not many big species in there, which is why we're focusing on the invertebrates and the microbes too, because it's quite easy to move them from one side to the other. But yeah, that's the question.
Okay.
It's a little bit difficult to relate what I'm thinking about to yours, but a number of us spent Saturday planting trees on a farm out near Gunning. And I just planting them in rib rows. I'm just wondering if you could relate what you're doing to that, perhaps by putting leaf litter around each individual plant, so that you could to create diversity, I suppose, going from plant to plant, because they're 4 or 5 meters apart.
Yeah, that's a good point. I think the thing that would benefit most from that is things like mycorrhizal fungi. So if you transplant leaf litter from a remnant site to the base of this plant, you might be introducing a lot of beneficial fungi for that specific plant. But I think this idea of leaf litter transplants is pretty useful in these sort of young revegetation sites, because the leaf litter isn't there, so it hasn't built up at all yet. So there's like a whole area that is essentially not habitable by all these litter dwelling invertebrates and microbes. I have people raise questions about, should you be taking this much leaf litter from remnant sites? So doing it on like a really massive scale might be difficult, but the amount that we take, find that doesn't really matter too much, because there's a really big turnover for these species. So you take a bit of their habitat and move them into another area, it's not too bad. But for doing it on like a broad sort of covering a whole revegetation plot, it might be a bit more difficult, I think. But yeah,
I was thinking just a patch.
just a little patch around. Yeah, I think that'd be a massive benefit, because there'll be a lot of insect species too that can go and sort of recycle the nutrients back into that soil surrounding that plant directly. So even at my house, we have all the leaf litter left over from the Tolgren funnels, and I'd like to take it home and dump it into my garden to do like a little transplant event essentially. And I find that little sort of species of plant pop up too, because we're taking the seed bank technically, so the top 5 millimeters of soil will take as well. So I think like a lot of these little remnant forest species have started popping up in my garden. So I've done transplants around plantings actually. Like I tried planting like a native garden at my house and mulched it with leaf litter from these remnant sites. But yeah, it's a question.
Thanks.
Well done. Oh,
I'll just read it out.
Okay.
Do you envisage increasing biodiversity as being beneficial to crop growth?
To crop growth, yeah, I reckon. I mean, the thing that I can think about the most is in the leaf litter layer, there are a lot of larval species of like flies, like little midges and little wasps as well. So if we're increasing the diversity of those larval species that go on and turn into adults, if they're close to like a crop, a canola or something that needs to be pollinated, having a greater diversity of these larval species would be beneficial to like immediately adjacent cropland, I'd say. Yeah, for sure.
Okay, the next one is from Adrian. Would you expect the soil profile to improve in depth? Would you expect the soil profile to improve and depth increase?
Yeah, I'd say so. If we're transplanting a lot of microbial species, they're quite important for like aggregating particles of soil. So that's something that can improve soil health. All these big invertebrate species, once they eat leaf litter, defecates, and that bit of defecation is kind of like a soil particle. So that's also good for the soil profile, forming these sort of aggregates of soil. I don't know about increasing soil depth. I imagine if the leaf litter is breaking down faster, there's probably going to be a thicker bit of soil in contact with that leaf litter. But yeah, I'm not entirely too sure about that. I've been focusing mostly on the leaf litter, not the actual soil. But these two are like obviously very connected with one another.
Okay, that's it. No more on the chat line.
all right. Fantastic. Yeah,
That's good.
very interesting. I'm sure everybody's go home and think about more questions. Good about that, probably. Yeah, that's great.
There's a lot of stuff to cover.
Yeah. So, thank you, Peter. And I'd like to call on Liz to offer a formal vote of thanks from Hartley Floor and Fauna.
Thanks, Peter. It's really rewarding for the OFF members to see where the research grant money is going. And it was very good of you to come up, I think, all the way from Melbourne to give this talk. Thank you. So we really appreciate the time you put into it. It's as Graham mentioned, a number of OFF members have been helping with regeneration planting in farmland areas. So this aspect, which we put the plants in, it's probably something we've overlooked a lot, is to the importance of the invertebrates, the microbes that accompany that revegetation. So this is already a wake-up call to us. Personally, I saw insects on Wednesdays as a volunteer at the Australian Museum. So honey beetles, brown beetles, and road beetles. That's really fun to hear that. Because they may not be so photogenic as the mammals, but they're so important, as you say. It's such a big part of the whole ecosystem and something we need to be reminded of. And I think finally, but your introduction about your work in Sturt was interesting, of course, because a number of our members have been recently out to Sturt, and the desert, we've heard from Richard Kingsford's group too. So yeah, building up an understanding of the desert area and then the agricultural area in Eastern Australia, it's really, really helpful. So thank you very much. And there's a small token, this is our annual calendar. There are a few photos of insects.
Oh, good.
You might have
I wouldn't accept another one. Thanks so much for that. That's lovely. Awesome. Cool.
Peter, we're going to move on to our general business section. You're welcome to stay, or you're free to go. Scoot off. Yeah. Have a something to eat. Yeah, steal your.
We don't want to. Well,
If anyone has any other questions that they want to ask, just pass on my email.
Yes, okay. We'd love to hear from you.
Yeah.
In that respect,
Yeah, for sure.
Anything we can direct people to think.
Yeah.
Yeah. We'll put the newsletter, we'll put out a monthly newsletter.
Yeah.
That's fine.
oh, yeah, still have PowerPoint slides. Session's still going, I think. Oh, try again.
All right. It was lovely meeting you. I'll catch you.
You too.
Yeah, good. You don't do this on the order. I'm going to stop recording too.
Yeah.